I want you to imagine, just for a second, that you're standing in your kitchen.
Okay.
You're prepping dinner, maybe getting a chicken ready to roast. And you turn on the faucet, you rinse your hands, and a single, completely microscopic droplet of water splashes off the sink and just lands squarely on your kitchen counter.
Right. And you don't even see it.
Exactly. It's entirely invisible. You wipe it with a sponge later. Maybe you tap that sponge against a plate. And then, like 2 days later, you are struck by a case of food poisoning so intensely painful, you're convinced you must have accidentally eaten raw meat.
But you didn't.
No. That single invisible splash was the sole vector.
It is a really sobering scenario.
Yeah.
And, when most people think of severe poultry-related illness, their minds immediately go to salmonella, right?
Oh, yeah, 100%.
It commands all the public health attention, all the warning labels on the packaging.
Salmonella definitely dominates the headlines. But our mission for this deep dive is to actually look at the pathogen that frequently outpaces it. So we're digging into a stack of medical, historical, and microbiological sources today to explore Campylobacter, and specifically Campylobacter jejuni.
Which is just a massive driver of bacterial diarrheal illness worldwide.
Yeah, but it operates almost entirely under the public radar. You don't really hear about it.
You really don't. And we're going to find out today that this organism's story extends far, far beyond a typical gastroenterology case. I mean, it is a defining case study in complex clinical neurology, global agricultural policy, and just the frustrating reality of how a pathogen managed to evade detection by the world's top microbiologists for the better part of the century.
The evasion aspect is where the history in these sources becomes genuinely baffling to me. I mean, the timeline starts way back in 1886.
Right, with Theodore Escherichia.
Yeah, the same pediatrician who gave his name to the Escherichia in E. coli. He was examining stool samples from children who had died of cholera infantum, and he documented seeing these very distinct spiral-shaped bacteria under his microscope.
He called them spirulum, I believe.
Right. So he knew they were present. He could physically see them with his own eyes, but the trail essentially went cold for decades because he just couldn't culture them in a lab. So what was the physical barrier actually stopping these early researchers?
Well, it was an environmental barrier. I mean, they were trying to apply standard aerobic culture techniques to an organism that simply could not survive them.
Oh, wow.
Yeah. So jumping ahead to 1913, British veterinary researchers, John McFadden and Stuart Stockman, they managed to isolate similar curved bacteria from aborted livestock fetuses, naming it Vibrio fetus.
But even then, they just dismissed it strictly as a veterinary issue, right?
Exactly. It took until 1957 for Elizabeth King, she was an American microbiologist at the CDC, to notice that similar human strains, isolated from children with severe diarrhea, required entirely different temperatures to grow.
Okay, let's unpack this for a second.
Yeah.
Because they had access to incubators,
yeah,
right? And petri dishes.
Oh, yeah.
So if they knew the bacteria existed, why couldn't they just adjust the temperature and let it multiply like, E. coli or staph?
Because Campylobacter is microaerophilic.
Microaerophilic, okay.
Yeah, meaning it demands a highly specific, very narrow atmospheric window of about 3 to 5% oxygen, combined with elevated carbon dioxide.
Wait, why does that matter so much? Like, why is normal atmospheric oxygen, which is around, what, 21%?
Yeah, about 21%.
Why is that a problem for it?
Because normal atmospheric oxygen is actually toxic to this bacteria.
Really?
Yeah. See, Campylobacter lacks sufficient quantities of certain enzymes like catalase and superoxide dismutase. And most other organisms use those to neutralize reactive oxygen species. So if you expose Campylobacter to normal 21% oxygen, those reactive oxygen species cause massive oxidative stress, literally tearing the bacterial cells apart.
Oh, wow. So standard incubators in the early 20th century were essentially death chambers for it.
Death chambers, exactly.
That makes perfect sense. They were drowning it in oxygen.
So how did they finally isolate it from human samples? Because stool is already packed with fast-growing flora, right? It would just overrun the culture.
It would. The major breakthrough finally came in 1972 from a Belgian physician named Jean-Paul Butzler. He knew human stool contained this organism, but the E. coli would just blanket the petri dish before the Campylobacter could even start growing.
Right.
So he relied on a physical filtration technique.
The 0.65 micrometer membrane filter. I loved reading about this in the sources. When I was reading about his method, it reminded me of like a nightclub bouncer or actually using a colander to drain pasta.
Oh, I like the colander analogy. Yeah. You pour the water out, And the bulky pen noodles representing the E. coli and other large gut flora, they get trapped by the plastic. But Campylobacter cells are unusually slender, and they're highly motile.
Very fast.
Right. So they act like grains of sand in that colander, just washing right through the 0.65 micrometer pores and into the pure culture medium below.
And that physical separation changed everything. I mean, once Butzler published that filtration method in 1973, clinical labs could finally isolate the human strains routinely.
But even after they could grow it, they still didn't really know how to classify it.
Right, because they were still calling it Vibrio, basically lumping it in with the bacteria that causes cholera.
Just because, under a microscope, they share that classic gull wing curved rod shape.
Yeah, and what's fascinating here is that the reliance on morphological shape was a massive pitfall in early microbiology.
Just looking at how it looks, yeah.
Exactly. In 1963, researchers Marcel Siebold and Maurice Ferrand proved that physical appearance was completely deceiving them. They looked at the biochemistry instead. And these organisms do not ferment sugars like true Vibrio species do. Their DNA-based composition is entirely different. Not to mention that extreme oxygen sensitivity we just talked about.
Right, the microaerophilic thing.
Yeah. So Siebold and Veron argued for a completely new genus, naming it Campylobacter, which is derived from the Greek for curved rod.
Did that reliance on shape cause other major misclassifications in the bacterial family tree? I mean, the sources hint at a few.
Oh, absolutely. The shifting taxonomy didn't stop in the 60s. The bacteria responsible for human stomach ulcers, Helicobacter pylori,
Oh, yeah, very famous bug.
is actually classified as a Campylobacter species for years.
No way.
Yeah. It wasn't until 1989, when genetic sequencing and metabolic tools became more sophisticated, that researchers realized they had to redraw the tree again and move it to its own genus. Taxonomy is essentially a constantly evolving argument based on the tools we possess at the time.
So, by 1977, British microbiologist Martin Scarrow developed a selective culture medium heavily spiked with antibiotics to suppress that competing flora. Labs started using it globally, and the medical community suddenly realized Campylobacter wasn't just some rare anomaly.
Not at all. It was everywhere.
Right, it was this ubiquitous disease that we finally had the microbiological lens to see. But that brings up the big question. Where is this massive reservoir coming from?
Well, the primary natural reservoir for Campylobacter jejuni is the gastrointestinal tract of birds.
Birds.
Yeah, wild birds, turkeys, and especially commercial chickens. They carry it as a harmless commensal organism.
So it doesn't make them sick.
Exactly. It does not cause disease in the avian host at all.
Which is wild. And, the temperature dynamic we talked about earlier explains a lot of that compatibility. A chicken's normal body temperature sits around 42 degrees Celsius, which is about 107.6 degrees Fahrenheit.
Very warm.
Yeah, significantly hotter than the human baseline. And according to the data in our sources, 42 degrees Celsius is the absolute optimal growth environment for this bacteria.
It is basically biologically engineered to thrive in that hot avian gut. And when you factor in the realities of commercial poultry farming, the transmission mechanics are staggering.
Oh, the scale is just massive.
Right. And chickens exhibit coprophagic behavior, meaning they peck at droppings. So in high-density flocks where they share water lines and litter, introducing just a single colonized chick can result in the colonization of an entire commercial house in a matter of days.
Wow.
Yeah, by slaughter age, a vast majority of the birds are heavily colonized carriers.
Which practically guarantees its entry into the human food supply. But, okay, once it makes the jump from a bird to a human host, the relationship changes drastically, right? It stops being a harmless passenger.
Very much so.
It's a gram-negative bacterium, meaning it has that tough outer lipid membrane shielding it, but its physical shape is really its primary weapon for invasion. Here's where it gets really interesting in the texts.
Oh, the invasion mechanism is brutal. So the human intestinal tract is lined with epithelial cells, which are protected by a thick, highly viscous layer of mucus.
Right, to keep things out.
Exactly. Most pathogens either get trapped in that mucus or flushed out. But Campylobacter utilizes its single polar flagellum and that signature corkscrew shape to generate incredible torque.
Like a motor.
Yeah, it literally drills its way through the dense mucus layer to reach the vulnerable epithelial cells underneath.
It just breaches the barrier entirely. But the subsequent damage isn't just from physical burrowing, right? Once it accesses those epithelial cells, it deploys something called cytolethal distending toxin, or CDT. How exactly is that toxin operating on a cellular level?
CDT is a remarkably aggressive mechanism. It enters the host's epithelial cells and travels straight to the nucleus, where it induces double-strand breaks in the host's DNA.
It breaks the DNA.
Yes. And the host cell detects this catastrophic DNA damage and immediately arrests its own cell cycle to attempt repairs.
So it halts cell division entirely. The intestinal lining basically can't regenerate.
Precisely. The cells undergo massive distension, they swell up, and eventually die. And this cellular destruction, combined with the physical invasion, triggers an overwhelming inflammatory response from the human immune system.
Okay.
And this mechanism is critical to understand because it dictates the symptoms. Unlike cholera, which produces a toxin that simply forces your gut cells to dump water.
Leading to that severe watery diarrhea.
Right. Campylobacter actually destroys tissue. That severe inflammation is why the resulting diarrhea is frequently bloody, accompanied by intense abdominal cramping and high fever.
Which circles right back to the kitchen scenario we started with. The public health consensus across these sources is really aggressive regarding the widespread myth of washing raw chicken. Have you ever rinsed raw chicken under the tap?
Oh, people do it all the time thinking they're cleaning it.
Yeah, but rinsing poultry under the tap does not mitigate the bacterial load. It effectively aerosolizes the bacteria, splashing contaminated water droplets across your sink, your sponges, and your utensils.
And that aerosolization is particularly dangerous because of the pathogen's exceptionally low infectious dose. I mean, if you consume salmonella, you generally need to ingest millions of bacterial cells to overwhelm your stomach acid and actually establish an infection.
Millions. Okay.
But human volunteer studies, which astonishingly involved researchers actually paying volunteers to drink Campylobacter cultures, proved that the infectious dose for this pathogen is vastly lower.
Wait, paying people to drink it, that's incredible, but just how low are we talking for the infectious dose?
The data shows infection can occur with ingestion of just a few hundred organisms.
A few hundred?
Yeah.
That drastically changes the threat model.
Yeah.
You don't need to consume undercooked meat at all. If a trace droplet from washing a chicken lands on a cutting board, and then you, I don't know, chop a cucumber on that board later, those few hundred cells are more than enough to bypass your stomach acid and drill right into your gut.
Exactly. Trace cross-contamination is the primary driver of domestic cases for exactly that reason. The margin for error in food handling is practically non-existent with this bug.
The gut invasion sounds violent enough, but for an otherwise healthy adult, the acute gastroenteritis typically resolves within about a week, right?
Generally, yes.
But the most alarming revelation in this deep dive, for me at least, isn't the initial illness. It is the neurological crisis that can emerge weeks after the diarrhea has completely stopped and the pathogen has supposedly been cleared from the body.
Yeah, this is where it gets truly terrifying. We're talking about Guillain-Barré syndrome, or GBS.
Right.
In a specific, albeit small, subset of patients, anywhere from 2 to 4 weeks post-infection, they develop a tingling sensation in their extremities.
Like in their fingers and toes.
Yeah. And that tingling progresses into bilateral muscle weakness, eventually manifesting as an ascending paralysis. It moves up the legs into the torso, and in severe cases, it can paralyze the respiratory muscles.
So they can't breathe.
Exactly. And Campylobacter jejuni is recognized globally as the single most common infectious trigger for GBS.
The mechanism converting a cleared gastrointestinal infection into peripheral paralysis is just wild. The literature refers to it as molecular mimicry.
Yes.
It essentially amounts to a biological friendly fire incident initiated by structural similarities, like identity theft on a microscopic scale.
That's a great way to put it. Walk us through how the immune system actually makes that error.
Okay, so every bacterium has a specific molecular structure on its surface. For Campylobacter, that surface is heavily coated in complex sugar structures known as lipoligosaccharides.
Say that 5 times fast.
Right, lipoligosaccharides. And during the acute gut infection, the human immune system maps those sugars and generates highly specific antibodies designed to seek out and destroy anything displaying that exact structure.
Which is the intended, healthy immune response to clear the infection.
Exactly. But the fatal flaw is structural overlap. The lipoligosaccharides on the bacteria are molecularly nearly identical to gangliosides.
And gangliosides are what?
They are lipid molecules that densely coat the surface of our own peripheral nerve fibers. So the infection is neutralized, the bacteria are totally gone, but your bloodstream is still saturated with these aggressive antibodies.
Right. And if we connect this to the bigger picture, those antibodies cross the blood-nerve barrier, they encounter the gangliosides on the peripheral nerves, and they just fail to distinguish between the host tissue and the pathogen.
They think your nerves are the bacteria.
Exactly. They bind to the nerves, initiating a full-scale autoimmune attack that strips the protective myelin sheath and damages the axons. The electrical signals from the brain can no longer reach the muscles, resulting in paralysis.
It completely shatters the notion that this is merely a transient stomach bug. You're dealing with a pathogen capable of triggering your own biology to dismantle your nervous system.
And because the clinical stakes range from severe bloody diarrhea to mechanical ventilation due to paralysis, the management of this pathogen has evolved into a major policy battleground. It really integrates agriculture, environmental science, and human medicine.
Let's examine the environmental transmission first, because the sources are very clear that poultry isn't the sole vector. There was a major epidemiological event in 1978 in Bennington, Vermont, that really shifted the public health perspective.
Yeah, the Bennington outbreak proved the vulnerability of municipal infrastructure. A town water main fractured, allowing contaminated surface water, which was likely carrying agricultural runoff, to breach the drinking water supply.
And what happened?
Consequently, an estimated several thousand residents contracted Campylobacter enteritis simultaneously.
Several thousand, just from water. So a single infrastructure failure resulting in thousands of infections completely validates the environmental transmission route. You don't have to mishandle a chicken to be exposed. You just need compromised water.
It cemented the necessity of the One Health framework.
What's that?
One Health is the epidemiological principle that human health, animal health, and environmental ecosystems are inextricably linked. The actions we take in one sector invariably cascade into the others.
Right, they all impact each other. And the most controversial collision within that framework occurred over antibiotic resistance in poultry farming. So what was the economic logic driving those agricultural practices?
Well, industrial poultry farming operates on razor-thin margins and massive scale.
Well,
To protect the flocks from respiratory diseases and maximize meat yield, it became standard practice to administer powerful antibiotics prophylactically via the birds' drinking water.
prophylactically, so giving it to them before they even get sick.
Exactly. And specifically, they utilized a class of drugs called fluoroquinolones.
Fluoroquinolones, that includes drugs like ciprofloxacin, right? Which is a critical heavy-duty antibiotic used to treat invasive human infections. Why would they deploy a human-grade antibiotic class in agriculture?
Simple economics, because it was highly effective at keeping the birds alive and growing. However, by dosing millions of chickens, they inadvertently created a massive evolutionary training ground for Campylobacter.
Because it kills off the weak ones.
Yes, the fluoroquinolones killed off the susceptible bacteria in the avian gut, but the rare, naturally mutant strains that possessed resistance, they survived. With no competition left, those resistant superbugs multiplied rapidly.
And because of the flock density and that coprophagic behavior we discussed earlier, those highly resistant strains just swept through the commercial populations, transferred to the meat during processing, and entered the human food supply.
Exactly. When humans subsequently contracted these resistant strains, clinicians found that standard fluoroquinolone treatments were failing. The drugs just didn't work anymore.
Wow. And this friction culminated in a monumental regulatory decision in 2005. The U.S. Food and Drug Administration officially withdrew approval for the agricultural use of enrofloxacin, which was the primary poultry fluoroquinolone.
Right.
And the sources emphasize how unprecedented that was. A federal agency actively pulling a highly lucrative veterinary drug off the market for the explicit purpose of preserving the efficacy of human antibiotics.
It remains a defining precedent in the ongoing battle against antimicrobial resistance. It forces us to acknowledge that the agricultural ecosystem directly dictates clinical outcomes in human hospitals.
But even ignoring the resistance issue for a second, there is a fierce debate among clinicians in the sources regarding whether to administer antibiotics to human patients at all. I mean, if a patient presents with severe cramping and bloody diarrhea, the natural inclination is to prescribe a pharmaceutical to stop it, right?
Yeah, and this raises an important question for doctors. It is a really difficult clinical dilemma, because for the vast majority of immunocompetent adults, Campylobacter enteritis is a self-limiting infection. The immune system will eventually clear the pathogen without intervention.
So what's the treatment then?
The standard of care is largely supportive, aggressive hydration and electrolyte replacement.
But wait, if the patient is suffering, why withhold an antibiotic that could potentially accelerate clearing the bug?
Beyond the macro-level concern of driving further antibiotic resistance, there are direct risks to the patient themselves. Clinical evidence indicates that administering certain antibiotics can paradoxically prolong the duration that the patient sheds the bacteria in their stool.
Oh, so it keeps them contagious longer.
Exactly. Furthermore, broad-spectrum antibiotics further decimate the patient's native gut microbiome, which is already under severe stress from the Campylobacter, complicating the overall recovery process.
So the consensus is basically to reserve intervention for the highest risk population.
Precisely. Clinicians typically restrict the use of targeted macrolides, like azithromycin, strictly for vulnerable groups. So infants, pregnant women, immunocompromised patients, or individuals presenting with systemic symptoms that threaten to move beyond the gut.
Right, into the bloodstream.
Yeah.
No.
For everyone else, time and fluid management remain the optimal approach.
So what does this all mean? As we synthesize the research from this deep dive, the trajectory of our understanding is just incredible. We started in the late 19th century with Theodor Escherich observing a mystery spirillum that frustrated science for decades.
Because it wouldn't grow.
Right. It required understanding the precise enzymatic limitations regarding reactive oxygen species and, deploying a 0.65 micrometer filter just to isolate it.
And once we possessed those tools, we uncovered a pathogen perfectly engineered to exploit the avian gut, utilizing a flagellar motor and a cytolethal toxin to breach human defenses with an infectious dose of mere hundreds.
It is an organism that dictates agricultural policy and remains a leading cause of bacterial diarrhea globally. But as we close out, I want you to consider the neurological aftermath we discussed with Guillain-Barré syndrome.
The molecular mimicry.
Yeah. If a brief, completely resolved gastrointestinal infection triggered by an invisible splash on a counter can initiate a case of molecular mimicry so precise that it tricks your immune system into paralyzing your own nervous system weeks later. How many other enigmatic chronic autoimmune diseases in the population today might simply be the biological friendly fire resulting from minor, forgotten infections that we just lack the diagnostic tools to see?